Good evening, everyone. I'm going to present my research issue on the transgenic potato conforming field resistant to light blight, but we have the difficulties in generating food safety data of pure RB protein. That is the issue, the big issue in an RB potato. That I'm going to tell in the very briefly what we generated since the 10 years data. So this is as we know that the potato light blight is one of the global challenging disease in the worldwide, since this Phytophthora infestation is such a difficult and effector biology is very, very difficult to understand because it is evolutionary changing its genome biology every once in 5 years because its genome is containing 70% of genome is containing the transposon elements. It's very difficult and that's why it's evolving its genome biology and the effector biology is breaking that resistant level. But fortunately, with the help of ABSP2 project, we got this line from the Wisconsin University as well as Gene Construct for developing this late plate resistant in our Indian potato cultivar. So we bought this line from the Wisconsin as a single transgenic event from the Wisconsin University as well as Gene Construct. But through introgression method, we got this positive lines based on the permission from the RCGM in 1999. We checked this single event, transgenic event in the field level. We got the field resistant. After that, we went for the introgression in that single event transgenic in our Indian potato cultivar, the popular that is Kufri Jyoti by backcross method. This is the flow of event I'm showing here. We selected that SP951 as the parental line, that is transgenic event carrying RB gene crossed with the Kufri Jyoti. And we continue successive generation of this screening as well as in the containment facility. And based on that yield potential, we got that, you can see that the control line is showing the bond of any green leaves is not at all seeing. So we selected the best 5 lines based on the continuous successive generation of around 7 years. So we selected these 5 lines based on the yield potential as well as the late blight resistant that we consider as a stable line after 7 years. So we checked that every year these lines as a stable line. Why? Because we checked this bioefficacy in terms of late blight every year under containment facility as well as in greenhouse. So because the Shimla is the very high disease pressure for the late blight, is the very, very congenial for the late blight. Every year we are checking under containment facility those 2 lines, all the 5 lines, and we checked the integration through southern hybridization. So we got that the same single event is present in our Kufri Jyoti also. So that for the, to know the site of integration between the event and the lines, we generate the flanking sequence. It doesn't affect any potato genome biology of our lines as well as in the event the SP951. So we studied that bioefficacy in terms of stability of the gene. How we checked that? We did that MAMA PCR assay. That means we have to generate the mismatch amplification mutation assay in that RB gene such a way that it will differentiate the endogenous R genes as well as our transgene. So we confirm that stability in terms of expression semi-quantitatively as well as stabilator marker like RB, any R genes, it is having some receptor. Actually, we didn't know that. After that, we checked that stability into upstream of the RB gene. That is called as SKP1, HSP90. Those are all involved in the stability marker for the RB. That's why our line is stably standing well till date in the field level. So come to the biosafety level, ultimately they will ask the pure protein studies for the toxicity. It's very, very difficult to generate the protein toxicity study because all RB genes, other than other R protein also, it is very intractable in nature. It is difficult to express their protein, whether it is in E. coli, bacteria, and the baculoviruses. So that's why it is very difficult to isolate the RB protein either from the transgenic event or from the wild species Solanum bulbocosum, which is originated from that wild species. So it is having several reports coming from the USA, Michigan State, in our lab also. We proved that it is very, very difficult to isolate the pure form of RB protein from our transgenic lines. So alternatively, with the help of ABSP2 project, Richard Goodman is generated that bioinformatic tools for this RB protein by using this Allergen online search database. So we generated that, we used that FASTA3 blast P of NCBA and all the site of allergen online version. So none of them showed the significant similarity with the more than 35% with the known RB protein. So finally we can say that, we can say that argued that regulated food safety and product cannot be undertaken due to its intractable in nature because it is supposed to be non-toxic.
Thank you.